Welcome to the deep dive. Our mission is pretty simple. We take a pile of complex research and, well, we pull out the insights that matter so you can get the full story fast.
And today, we're tackling something truly monumental. We are mapping a new spectrum, not of light, but of sound.
Right. We're diving into a breakthrough in biomedical imaging, specifically looking at how we can see gene expression deep inside the body using ultrasound.
It's the first real success in what's called multiplexed ultrasound imaging.
Okay, so before we jump into the new stuff, let's set the stage with the reigning champion. Right, fluorescence.
For decades, if you wanted to see what was happening inside a cell, you used green fluorescent protein,
GFP.
or one of its colorful cousins. And the resolution is it's incredible. But it has always had this one major Achilles heel.
It's basic physics, really. Light scattering. I mean, try shining a flashlight through a thick fog. The light just diffuses. It goes everywhere. Living tissue is even worse.
So you can only see things right at the surface. Exactly. Once you go deeper than, say, a single millimeter, your beautiful high-res signal is just, it's lost in the noise. It's gone.
Which is why ultrasound is such a critical alternative. Acoustic waves just behave differently in tissue.
Completely differently. They let us see several centimeters deep, not millimeters. And we can still get really impressive resolution down to about 100 microns.
So to make cells visible to ultrasound, scientists came up with these things called acoustic reporter genes, ARGs.
Right. And these genes, they produce tiny air-filled protein bubbles. They're called gas vesicles, or GVs. You could think of them as the acoustic version of GFP. They create a contrast signal wherever they're expressed.
Okay, so if these ARGs have been around, why is this specific paper a breakthrough? What was the big bottleneck?
It was what we call the single sound problem.
A single sound.
Yeah. Think about why fluorescence is so powerful. You've got red, green, blue, yellow proteins. You can track 4 or 5 different things at once because their light signals don't interfere. They're orthogonal.
I see.
Until now, ARGs could only produce one type of sound. So you could track cell population A, or you could track biological process B, but you could never track them both at the same time in the same animal.
So the big mission was to create more sounds, different colors, but for ultrasound.
That's the core of it. And the solution wasn't about changing the pitch or the frequency. It was about changing something much more physical.
Okay.
The breakthrough was figuring out how to tell ARGs apart based on their unique acoustic pressure response profiles.
So not how they sound, but how they react to sound.
Precisely. They tuned the rigidity of the protein shell itself, and that's how they developed the first two-tone ARGs, BRG560 and BRJ710. They basically gave ultrasound its first green and magenta channels.
Okay, let's unpack that because that's where the physics gets really cool. How does a tiny protein bubble even create a signal for an ultrasound machine?
It's a really clever mechanical trick. Imagine a gas vesicle as a tiny, hollow, air-filled can made of protein. When an acoustic pressure wave from the ultrasound probe hits it, it doesn't just vibrate. It does something called reversible mechanical buckling. It momentarily collapses inward and then snaps right back to its original shape.
Oh, like when you pop the little metal cap on a Snapple bottle, it takes a certain amount of force to push it in, and that pop is the signal.
That is a perfect analogy. And that snap is what gives us what we call nonlinear ultrasound contrast.
Nonlinear. What does that mean here?
Well, the sound wave you send in is at one frequency, let's call it F1. If the bubble just vibrated, the echo coming back would also be F1. But because of that sudden violent snap, the returning echo contains harmonics.
Like overtones in music.
Exactly. Frequencies like 2F1, 3F1, frequencies that weren't in the original signal. So we could just filter out all the normal tissue echoes and listen only for the sound of those snaps.
Which means if you can change how much pressure it takes to make that bubble snap, you can create different signals.
Now you've got it. That's the key. The researchers went after a protein called GVPC. This is a structural protein that basically acts like scaffolding on the GV shell, and it dictates the stiffness.
So tweak GVPC, and you can tune the pressure threshold.
That was the hypothesis.
So walk us through the first one they made, BARG560. They wanted a softer shell, one that would buckle under lower pressure. How'd they do it?
They started with an existing system called BARGseer. To make the shell softer, or as they put it, more permissive, they deleted the GVPC gene entirely.
Just took it out.
Yep. The logic was, if you remove the main stiffening agent, the shell should get softer.
And did it work?
It worked spectacularly. The original version had a pressure threshold of about 820 kilopascals. By removing GVPC, that threshold plummeted down to 560, and that became BRG 560.
A lower threshold is great, but did making it softer make the signal weaker? Did the snap get quieter?
That's the most amazing part. No. The signal got louder, a lot louder. The maximum signal for BRG 560 was almost 6,000 units, compared to only 1,000 for the original. They didn't just create a new channel, they engineered a contrast agent that's 4 to 9 times brighter.
Wow, so that means you can get a clearer picture faster.
Way faster. It's a game changer for imaging in a live animal.
Okay, let's do a quick recap here. We're using gas vesicles, or ARGs, for deep ultrasound imaging. The signal comes from reversible mechanical buckling, which creates a clean, nonlinear contrast.
Right.
And by deleting the GVPC stiffening protein, the team engineered BRG 560, which is super sensitive, works at low pressure, and is incredibly bright.
Which brings us to the much harder problem.
Yeah.
You have your soft, loud signal. How do you engineer a second, distinct signal? One that's stiffer, so it has a higher threshold, but not so stiff that the pressure you need to trigger it accidentally destroys your first signal.
Right, and that brings us to engineering the second tone, BRG 710, and the math behind making it all work together. The goal was a threshold at least 100 kilopascals higher than BRG 560, but they had a hard ceiling they couldn't cross.
A very hard ceiling. That was the collapse pressure of BRG 560 itself, which is about 1,220 kilopascals.
So if you push too hard, you don't just get a snap, you permanently crush the bubble.
Exactly. The whole game was to find a second tone that worked inside that non-destructive imaging window, somewhere between 560 and 1,220 kPa.
Okay, so just deleting GVPC made it too soft. They needed to find a more subtle way to adjust the stiffness.
Right. So they turned to a technique called directed evolution.
So they're not designing it, they're breeding it.
In a way, yes. They took the GVPC gene and intentionally introduced random mutations into it using a process called error-prone PCR. They generated 879 different versions of that stiffening protein.
And then they tested every single one.
Every single one. They mapped each variant based on two things: its pressure threshold, the P0, and its maximum signal strength, the S-max.
What did that map of 879 variants tell them?
It revealed a really fundamental trade-off in the physics of these things. Strong negative correlation. It means the universe of acoustic proteins follows a simple, kind of ruthless rule. You can either be hard to buckle with a high pressure threshold, or you can be loud when you do buckle with a high signal.
But you can't be both.
You cannot be both. Stiffer shells need more force, but that very stiffness limits how much they can deform, which makes for a quieter snap. They were searching for the Goldilocks zone.
And which mutant was their Goldilocks, the one with that perfect intermediate stiffness?
The winner had a single amino acid change, L154P, and this new construct became BRRG 710 with a nice, distinct threshold of 710 kilopascals.
Just one amino acid.
Just one. And when they ran structural predictions, it looks like that one change introduces a little kink into the protein structure, which subtly changes how it binds to the GV shell, and that was enough.
Okay, so now we have two signals. BRG 560, the soft and loud green, and BRG 710, the slightly stiffer magenta. But when you look at a mix of cells, the 710 signal only turns on after the 560 signal is already active. How on earth do you separate those overlapping signals in the same pixel?
This is where the math comes in, and it's called linear unmixing.
Okay.
It's a really clever bit of linear algebra. First, you have to create a calibration key. You take pure samples, one of just BRG 560 cells and one of just BRG 7 and 10 cells, and you image them across the entire pressure range, 500 kPa, 600, 700, all the way up.
So you're building a perfect reference profile for each one.
Yes. That reference profile becomes your multiplexing matrix, or M. Then you image your unknown mixed sample across that same pressure range.
And then the unmixing happens.
Then, on a pixel-by-pixel basis, a computer solves an equation to figure out how much of the signal in that pixel came from the green reporter and how much came from the magenta one. It separates them out into two clean channels.
That sounds computationally intense. And is there a risk of crosstalk? I mean, couldn't the algorithm get confused since the pressure ranges are so close?
That's a great question, and it's a calculated risk.
Yeah.
But the precision of that initial calibration matrix, M, is what minimizes it. Because they map the entire response curve, not just the on-off point, the algorithm is really good at predicting and subtracting out the overlap.
And they proved this works in a dish first, right?
They did, in two really cool ways. First, they showed they could tell two different types of bacteria apart. They put BRG 560 into Salmonella and BR 710 into a probiotic E. coli. When they mixed them, the ratio of the green to magenta acoustic signal almost perfectly matched the actual ratio of the cells they put in. It was incredibly accurate.
Wow. And the second application?
The second was even cooler, I think.
Right.
They showed they could track a cell's internal state. They engineered the E. coli with a genetic switch. Normally, you would make the green BRG 560, but if they added an antibiotic tetracycline, the cell would flip a switch and start making the magenta BRG 710 instead.
So they could watch the cells change their minds acoustically.
That's exactly what they did. They added the trigger, and they watched the whole colony shift its signal from the green channel over to the magenta one. It's proof you can track internal programmed events.
That is some really solid groundwork. So we've got the engineering, we've got the math now for the big test. How does this beautiful system handle the messiness of a living, breathing mouse?
Right, because moving in vivo introduces all sorts of real-world problems. You've got background noise, you've got sound waves weakening as they travel through tissue.
So let's start with their first in vivo test, tracking bacteria in the GI tract. That's a notoriously difficult place to image.
It is. So they transplanted the two bacteria, the STM560 and the ECN710, into the colon of mice. And even with all the complexity of living tissue, the unmixing algorithm held up.
It still worked.
It did. The acoustic channels clearly separated the two populations. Mice that got just the salmonella lit up almost exclusively in the green channel. Mice with the mix showed both. They had successfully created two independent tracking markers for deep tissue.
That's incredible. But the real holy grail for this kind of tech is probably cancer therapy, right? Tracking therapeutic agents.
Absolutely. And that was their second big in vivo demonstration, tracking tumor colonizing bacteria.
So explain the concept there. Why would you want bacteria in a tumor?
Well, certain engineered bacteria, like Salmonella and the specific E. coli, have a natural tendency to colonize and grow in the low oxygen cores of solid tumors.
Okay.
Scientists are trying to harness that, turn them into little drug delivery factories right at the site of the cancer. But to do that, you have to be able to track them, see if they got there, see if they're still alive.
And with multiplexing, you could track two different therapeutic strains at once.
Or track an initial dose and then a booster dose you give a few days later. So they injected mice that had colon cancer tumors with either a single bacterial strain or a mix of both.
And then they brought in the ultrasound. What did the separated channels show them inside that tumor?
The separation was crystal clear. In the mixed samples, they saw strong signals in both the BRG 560 green channel and the BRG 710 magenta channel, deep inside the tumor mass.
And they could verify this.
They did. They followed up with histology, the old school method of slicing up the tissue and looking under a microscope. And it confirmed visually that both bacterial species were right there, co-colonizing the tumor core. The acoustic picture was real.
That is a massive proof of principle. But you mentioned challenges. What's the biggest limitation they ran into in big tissue?
The big one is acoustic attenuation. Sound just loses energy as it travels through tissue.
It gets weaker with depth.
Exactly. So that 560 kPa pulse you send from the probe might only be a 300 kPa pulse by the time it reaches a deep tumor, which means your BRG 560 might not even turn on.
And if the pressure isn't what you think it is, your whole calibration matrix, your M, is wrong. The unmixing algorithm fails.
Precisely. The math falls apart if the local pressure is unknown.
So how do you fix that?
They proposed a really elegant solution, a depth-corrected multiplexing matrix, or M2D. Instead of one master key for the whole image, you'd have a different key for every depth.
So you'd tell the algorithm, for pixels at 1 centimeter deep, use this pressure calibration. For pixels at 3 centimeters, use this other one.
That's the idea. It's like constantly refocusing a camera lens depending on how far away the object is. It's the path forward.
So connecting this back to the big picture, the strategy they have established here, tweaking that GVPC protein, that can be applied more broadly, right?
Oh, absolutely. The same GVPC protein is used in the ARGs being developed for mammalian cells. So this exact same approach could be used to track, say, 2 different types of cancer cells or 2 different populations of immune cells fighting an infection. We're moving from just 2 tones towards, well, a whole symphony of ultrasound signals.
This really feels like a monumental shift. Ultrasound is finally catching up to fluorescence in its ability to do multiplexing, but it has this incredible advantage of deep tissue access.
And, the historical parallel is what's so exciting to me. The entire field of fluorescence imaging started with one protein, GFP. And through this kind of engineering and directed evolution, it exploded into a whole rainbow of colors.
And we're at that same starting point now for sound.
We are at the very beginning of decoding a new, vibrant acoustic spectrum.
Which brings us to a question for you, the listener. We've now established that we can track where two different types of cells are deep inside the body. So now that this two-tone tool exists, what two distinct cell populations, whether it's in the gut microbiome or in a complex cancer therapy, what two things do you think scientists should prioritize tracking at the same time to unlock the next huge biological insight?
The body's deepest secrets are finally ready to be heard.
Thanks for diving deep with us.